welcome members, visitors across Australia and overseas. We acknowledge the traditional owners and custodians throughout this land and pay our respects to elders past, present and emerging. We also recognise the deep cultural and scientific knowledge of Australian fungi held for millennia by Aboriginal Torres Strait Islander peoples, and we welcome you here today. My name is Susie Webster and I thank you all for joining Fungi Maps Fungi lecture series. At any time during today's event, please feel free to submit your questions by clicking the Q&A button at the bottom of your window. And our hosts, myself and Sophie Green, will raise these for our speaker to answer during a brief interval in the middle and then again at the end. Our topic for today is Australian Boletes, from there to here. It is my great pleasure to welcome today's Fungi speaker, Dr. Roy Halling. He is Curator Emeritus of Mycology at the New York Botanical Garden, where he has carried out research in the classification, systematics, biogeography and diversity of mushrooms. Roy has been visiting Australia since 1992, I think. Correct me if I'm wrong, Roy.
Oh, that's right.
It's good, okay. And I will let you tell the rest. So Roy, your eager audience awaits you.
Thank you, Susie, and thank you, Sophie, for inviting me to give this talk. And there's a lot of people that I want to thank, but I'll wait till the end to do that. But something that you don't really begin to realize until you get down deep into studying something. And so I want to bring you up to date on how I got going on all of this stuff. And it takes us back a little while. Come on. There we go. When I was an undergraduate going to State College in California, I was really interested in plants. So I was trying to take every class I could that was botany related. And back then, fungi were considered plants, different kinds of plants. And the professor who taught the mycology class, he was a specialist in marine fungi. And what he did his PhD on was an obligate parasite that grew on marine crab eggs of the king crab in Puget Sound near Seattle, Washington. So what he tried to do was to isolate this fungus, grow it in artificial culture, make it jump through its hoops so he could figure out how this thing would go through its life cycle. So what he did was, back in the day when digital were things that you did with your fingers and toes, he had a 16 millimeter film camera attached to his microscope. And what he did was he filmed the sporogenesis of this fungus that he presented in a film to the International Botanical Congress in Seattle in 1969. Now, there's something I want to point out to you. There's going to be a lot of scratches and things from this film. But as you see in the middle of this frame here, just to the left of that big vertical scratch, there's a blank, dark line. What it is, it's a piece of the cytoplasmic or the plasmalemma that's connecting cytoplasm to another packet of cytoplasm further to the right. So this video lasts about a minute, but it shows you what he was able to capture on film of the sporogenesis of this fungus. And it produces zoospores because it grows in water. So watch how it goes. The thing that I want you to pay attention to is what's powering the cytoplasm moving into that vesicle on the left. Is it being pushed or pulled? It doesn't appear to be either of those things. And as far as we know, nobody's been able to explained the physics behind the cytoplasmic flow in this, what was called a fungus back then. Based on what we know now, this is not truly a fungus in the DNA sense. But once all the cytoplasm comes up, then it's like cats in a bag, and they're fighting, and the zoospores are released from the vesicle at the end of that discharge tube. So when it ends, I'll move on to the next slide. So I asked Dr. Gottelli, I said, well, what do we know about fungi? And he says, we don't know anything about fungi. And everybody in the classroom goes, what are we doing here? And so we decided, okay, we'll have to explore this in the class, which we did. But some field trips that we did in Central California in the mountains of the Sierra Nevada, we were finding boletes. And I asked Dr. Gottelli, I said, what are these boletes? And I said, I don't know. And I said, well, you're a mycologist, you should know. And he says, well, if you want to know, you should go to grad school. Okay. And so I did. I went to San Francisco State to study with Harry Tiers, who was a premier bolete specialist at the time. And I learned a lot about my boletology from Harry Tiers. And some enigmatic things that grew in California at the time was this one bolete down on the lower left called Boletes eastwoodii, named for a woman who worked on plants at the California Academy of Sciences in San Francisco. Luckily for me, then I had a chance to describe my first species, which I call Boletes citrinoporus. So the 2 years that I had in California studying boletes was 1974 to 76. After that, after my master's degree, I went on for a PhD, did a postdoc at Harvard, and then moved on to the New York Botanical Garden in the early 1980s to take up a post as Curator of Mycology. When I was there, I got a letter from Nick Malachuk, who was working for CSIRO in Perth. And he invited me to come along on an expedition in Queensland in 1992. And so I said, heck yeah. And I came. And I met up with a group of folks from left to right there. That's Mike Castellano from Oregon, Jim Trappe, and Neil Bowser, then Ben Bayless, Paul Riddell with his back to us, and another chap named Roy Young. And so we had a chance to go up north of the Daintree River in northern Queensland to collect fungi, and elsewhere in Queensland. We also had the opportunity to go to Papua New Guinea to do some work there. The primary purpose was to collect macrofungi that were associated with gum trees and to see what kind of mycorrhizae were associated with those gum trees. Subsequent to that trip, turns out that a genus was named for Mike Castellano called Cassia. Jim Trappe had a genus named after him called Trappia. Neil had one named Bowsera. Paul Riddell had one called Riddellomyces, and Roy Young had one called Roy Youngia. And I thought later on, I said, one day I got to get back here and study this stuff. So in 1992, I had the chance, as you can see on the map, I had a chance to be in Northeast Queensland and Papua New Guinea. But it wasn't until, 13 years later, I had a chance to get back to Australia to figure out what the heck I was seeing in the field in 1992. So in those 15 years, 2005 to 2020, I had a chance to visit several places in Australia, New Zealand, Indonesia, particularly in Java, and in Thailand. and try to get my mind around what it was I was seeing while I was working on boletes. One of the things that you have to do if you're going to pick up and work on a group of mushrooms or any group of organisms, you have to understand what was published in the past. In Australia, a lot of the macrofungi were published by three guys anyway, who lived in Britain. One was Miles Berkeley, Mordecai Cook, and George Massey. And Mordecai Cook wrote this handbook of Australian fungi, I believe it was in the late 1800s, early 1900s. And so try to understand what it was they were describing that of material that was sent to them. And back then, the large quantity of things, of material that was sent to them was sent by Ronald Gunn from Tasmania, von Mueller from Victoria, Bailey from Queensland, and I'm not exactly sure if Rodway sent material there, but he was also instrumental in looking at Tasmanian macrofungi. Since then, John Cleland and John Cribb worked a lot on macrofungi and some boletes in Australia. But as I said, going back to try to understand what it was that was being described at the time. And here's an example from the mid-1850s of a specimen that Ronald Gunn sent to England, and Berkeley described it in one of his publications, and he called it Boletes fruticicola. Well, if you look at the specimen that Berkeley had in front of him, it looks like a piece of tar or, bitumen off a road, and it's glued to a piece of cardboard. And it's extremely hard. If you try to cut a section with a razor blade, the edge of the razor blade will curl up because that fungus tissue is so hard. And you look at the description and you try to figure out what it is they were actually describing. It's very, very difficult just based on the Latin description and the English description that follows it. The other thing, pay attention to here, it says hat down in this part of the description here, it says hab penguite. Well, it took me forever to figure out what penguite was. Well, there's no penguite in Tasmania. If you look at the original label, that's a Q. So the locality where Gunn found it was Penquite. So there's a typo. So that's part of the forensics of figuring it out, what you have and where you go. It wasn't until the middle 1980s, Roy Watling at the Royal Botanical Garden in Edinburgh started to look at boletes in Australia. And he started authoring a bunch of papers that I call the Colula papers, beginning in 1986 through 1999, describing boletes, as he says, boletes of the Colula sand mass. And it wasn't just boletes from Colula in Queensland, but he also described a lot of things from elsewhere in Australia. But one of the points that I recognized in one of the introductions of one of the papers, that a particular bolete genus, Tylompilus, is well represented, and there's heaps of species left to describe. And so that's 13 years that the other Roy spent writing about boletes in Australia. At that time, and while I was growing up, the generic concepts of boletes was quite variable depending on how you wanted to group species. So Smith and Tears, for example, were, lumpers, as we say, with about 12 species, or 12 genera, in the that they recognized in their book, landmark book called Boletes in Michigan. But over 4 decades, Rolf Singer had expanded that concept, generic concept, to include about 25 genera. And so depending on which book you wanted to read, you could apply generic concepts to different species depending on where you were. Once it was discovered that boletes had DNA, the family Boletacea, in the sense of Rolf Singer, got carved up into 4 additional families, which were placed in 3 suborders of the order Boletes. And this was back in the middle 2006 in a landmark paper published by Manfred Bender and David Hibbett. But in late 2013, early 2014, what I call the DNA apocalypse, when gene sequences were used to recognize different taxonomic groups of boletes, we're now up to 87 different generic hypotheses just based on DNA data. And what Wu et al. Did was to divide up the Boletaceae in the sense of the the Bender and Hibbett classification into different subfamilies. And one subfamily called the Boletes group didn't hang together too well in terms of statistical support. But at that time, a lot of truffles, or have been called sequestrate fungi, which were formerly known as gastromycetes, turned out to be boletes. And so those that are marked with an asterisk are the sequestrate or truffle-like things that are actually boletes. Based on what I know and over the last decade or so, I could recognize about, out of that 87, I can recognize about 22 genera that exist in Australia that I have seen, or ones that have been shown to me either in a photograph or a dried specimen. And the number of species, minimum, is about 100. And there's a lot more. Many have not been described yet. But these are the ones that I think are natives, at least so far, that we know about in Australia. And this is sort of a montage of what they look like and their generic names. Now, this isn't everything, but it's the ones I've got good pictures of. Boletes is one of those. And most recently, past December, Nigel Fechner, Naveed Davudian, and Teresa LaBelle were able to describe this iconic thing that we know that occurs in Queensland, at least from up the Atherton Tableland all the way down to at least as far as the border ranges where Queensland meets New South Wales. And it has this distinctive rim here, which is attached to the edge of the pileus when it's very young, and it splits. And you can see the ornamentation on the cap matches that on the stipe, but above that, it's completely naked. So and based on DNA data to support it, it was a new genus that we call Boletes. And I mentioned the truffle-like sequestrate genera that DNA's told us were related to the boletes. The ones indicated with the blue arrows there haven't been reported from Australia yet, but it's possible that they could show up because they're up in Southeast Asia, and it's possible that they might have crossed Wallace's line and show up at some point in Australia. It's just not known yet. But this is what they look like. Durianella up there in the upper left and Spongiforma on the lower right. And Royangia there in the middle, that's the one that was named after the guy that was on the boat that we were crossing, or the ferry boat crossing the Daintree River. So that truffle bears his name. And there's other ones. And Teresa Labelle is the guru on these kinds of truffle-like fungi. And so without her input, Octaviania, Rossbeavera, we wouldn't know anything about those particular bolete truffles. So how do boletes pay the rent is one of the things that have interested people. And the bolete lifestyle is essentially a obligate association with tree roots or plant roots that are called mycorrhizae. And it's normal, it's standard procedure for boletes. And sometimes it's difficult to actually ascertain which tree or which plant a bolete might be associated with, but we'll get to that in a second. But if we zoom in on that picture of the lower left of that root association, you can see that the tree in the middle only has 3 roots. The rest of that stuff in there is a mycorrhizal fungus. And the mycorrhizal fungus is connecting that one tree with the 2 others to either side of the heading there that says biological significance. And so what is this doing? I mean, the tree is actually, because of the fungus, has more access to soil volume, more access to water, minerals that the fungus transfers to the tree. In return, the fungus receives carbohydrates in the form of starch or glucose to allow it to live. So there's all kinds of selective advantages here for this kind of obligate association. And let me just show you an example of one that we explored when I was doing work between 1992 and 2005. I was working in Costa Rica, up in the alpine zone called the Paramo in the New World. There are no trees, but you have these tall shrubs that might be 3-4 meters tall. And when you're walking around in there, you can actually get into parts where you can actually get inside that shrubbery and walk around without having to crawl to look for fungi. Well, it turns out in this particular habitat, there was a bolete. And the right time of year in the rainy season, you could fill the back of your ute with this bolete. And I'm thinking, what the heck, because it's mycorrhizal, I assumed it was mycorrhizal. What is it growing with? There's nothing here that I know that is a mycorrhizal plant. Turns out there was a member of the blueberry family called Comarostaphylos arbutoides, and I had a grad student working with me at the time named Todd Osmondson, and what he decided to do was to dig up earth balls underneath the mushroom, then wash the soil away to get to the roots. And that's what you see in the upper right, is some roots from underneath the earth balls of this Lexinum. And if you make sections of the roots, you can see in the center, bottom center and the right, lower right, that the root cells are filled with fungal hyphae. So what Todd did was he sequenced DNA from the plant, he sequenced DNA from the mushroom, then he sequenced fungal DNA from the root, and he sequenced plant DNA from the root. And he took those sequences and he compared them. And as you see in those colored columns in the center there, the fungal DNA F matched perfectly from root and the plant DNA from the root and the plant matched perfectly. So clearly it was a mycorrhizal association that had not been documented before in the subalpine of Central America. So what's the significance of this? And it's something that I've called four-part harmony in a sort of humorous way. Back in the 1990s, there was mentioned an endangered bird called the northern spotted owl. And it was subject to conservation status because of a lot of lumbering that was going to go on in the forests where this bird lived. And the bird needed old growth Douglas fir forests in order to survive. It turns out that the main food source for this spotted owl was the northern flying squirrel. Well, the northern flying squirrel needed to eat something, so that rodent ate truffle-like fungi. Those truffle-like fungi formed obligate mycorrhizae with the Douglas fir. So we have a bird icon for conservation, but the basis for maintaining that ecosystem was the fungus. It kept the tree alive, it kept the rodent alive, it kept the squirrel alive, which fed the spotted owl. The trees staying alive had a place for the spotted owl to roost and nest. So how does this look in Australia? I think it's the same thing. You've got birds, carnivorous birds who will eat bettongs or potoroos, and they have to have a place to live, so the potoroos eat fungi. The fungi form mycorrhizae that keep the gums alive. The gums are trees where the owls can live. So in a way, in a similar fashion, this sort of four-part harmony is also going on in Australia. And I'm not making this up. This has been shown by Andrew Claridge and others in Australia. So at this moment, I want to just take a break and take a breath. And if we have a minute to do some Q&A, that's cool. Susie,
okay.
Sophie?
Yep. Hi, everyone. Yeah, I'm Sophie from Fungi Map. Thanks a lot for tuning in today. We do have a question. So, Roy, are all boletoid fungi mycorrhizal?
There's a few that are not. For example, the one that I knew about from Fran Gard, she told me about this thing, and it's called Buckwaldo boletes. And it might be an invasive with pine, but it's a parasite on a wood rotter, polypore, that rots wood. And the other one that I know about is another invasive called Chalciporus piperatus. And it's associated with the fly mushroom, the Amanita muscaria. So when that came in with pines, not only did Australia get Amanita muscaria, it got Chalciporus piperatus too. So I got 2 for 1 on that invasion. So there's a couple of them that are not absolutely mycorrhizal. There's one genus, which is a parasite called Pseudoboletes, which is a parasite on sclerodermas. But as far as I know, that doesn't exist in Australia yet. But yeah, 9 out of 10 are mycorrhizal.
Excellent, thank you. And we do have one other question, might be a bit of a tricky one to answer. What do you think Australia might look like if we had no boletes?
Hey, geez, I don't know. That's a tough one. I have no answer, Sophie. Sorry.
No worries.
I'd have to go. I would have had to work somewhere else.
We've also got, how can you tell whether the DNA matches between the mycorrhiza and the plant?
You mean in the plant root? No,
Yep.
Okay, the way fungal DNA is of a specific kind. So you, when you isolate it, you can isolate specifically for fungal DNA that will exclude plant DNA. And they're called primers. And so when you extract the DNA, you can specifically pinpoint for fungal DNA, even if you pull it from a plant root. And the same thing for plants. You can pull specifically plant DNA and pinpoint for it. So that's how Todd did it when he was doing the root extractions. Does that make sense? Did I get it?
Beautiful. Yep. I've got two more questions, and then I might leave some more for later. Do you use iNaturalist as a tool for any of your research?
I haven't yet. I did it once for a leech I found. Sorry, it wasn't a leech. What was it? It was something I found in New South Wales walking around in it, and I didn't know what it was. And so I posted it on iNaturalist, and somebody came back and told me what this little slug-like thing was. Okay, great. But I haven't done that yet, no. I mean, I've posted all my data onto the New York Botanical Garden virtual herbarium website. So all my bolete data is there, and it's been uploaded into the Myco portal. I've made some, I think a lot of my data that's deposited in Melbourne or Brisbane or Perth has ended up in the AVH, the Australian Virtual Herbarium. And I think that, I don't know if that's tapped into by iNaturalist or not.
And we'll send people a link to your website as well that they can look at later. Okay. And finally, is edibility something you discuss or investigate?
As I've had people ask me, is this edible? And I said, everything's edible once. And a board member of the New York Botanical Garden asked me that, and she says, I think the proper question is this edible twice? And so as far as Australian boletes are concerned, I have no idea. I don't know that. I only know of one case of a poisoning of a bolete that happened in southern Queensland a while back, but I've never been able to look at the fungus to see what it was. But as far as I know, in other parts of the world, boletes won't kill you. They can make you sick, but they won't kill you. So I'm really wondering what that fungus was that was called a bolete that caused somebody to die in southern Queensland a while back.
Yep. I guess with everything related to fungi, there's always more research to be done.
Absolutely.
All right. Well, keep your questions coming, and we'll do some more at the end. I don't know if you need a quick drink of water or anything, Roy, before you keep going with your second half.
Yeah. Let me. Okay.
Thanks everyone for your questions.
Okay. Part two. All right. So instead of showing you every picture of every bolete that I ever have seen in Australia, let me just go through a couple of groups of things that we know something about. It doesn't mean that all of the species are described, but this is what we know, at least what I know up to this point. Okay, and so we're going to start in the upper left, go clockwise, and then once I finish those four groups, then I'll talk about some what I call newbies, things that we don't know yet, but they're out there, they're abundant, and no doubt you've seen them. Now, the first group I want to talk about is this thing called Bolete. It was a species described here in southern New York in 1912 by Charles Peck, and he named it for the collector whose name was Charles Balu. And it's this brilliant bittersweet orange bolete. And in Australia, you can find these brilliant bittersweet orange boletes. And a lot of some mycologists going back a couple of decades transferred a lot of these species into this genus called Rubinoboletes. It's not a Rubinoboletes. Rubinoboletes is a segregate genus from what we know is Chalciporus, based on morphology and based on DNA. And right now, we're working on this group to try to sort these out. So using the species name Balui, use it in the broad sense for a group, a concept, a species group, okay? So I just want to show you some examples of this Balui group, but they're not all this brilliant orange. And this example is, I've seen before, not just in Southeast Queensland, but they have the fine pores, they're orange, they can fade a little bit, but that's a classic one that shows up in Australia. On Fraser Island, where I had the chance to work with Nigel Fechner over a 3-year period, there's heaps of these things, but they get a little contorted and they form these clusters. And it seemed to me, and I think to Nigel too, that they were the most common bolete that we could find in the sand on Fraser Island. And the other thing about them was that they were often very difficult to get spore prints from. And what we kind of figured out was they were on their way to becoming truffle-like. So when we saw examples like this, we were assuming they were on the way to becoming a truffle, but hadn't got there yet. You couldn't get a spore print from these things. Up in northeastern Queensland, there's more of this orange thing, but the hymenophore, the tubes, are decurrent. The pores, tubes, they go down along the edge of the stipe, and they're very coarse and radially oriented. And when you get up close and personal to those pores and tubes, you can see the pores have pores, and then those pores have pores. So it's a multi-layered aspect to the spore-bearing surface, the pores of these boletes. In Colulas, having Watling, having looked a lot there, we had to go to Colula to see if we could find some of the same things that Watling did. And here we found a red cap form of this Balui group. Oops. On Fraser, we found a pink cap form, but with practically no stipe. And it's essentially the same in the same group. Up in the Queensland rainforest, up in off the Atherton tableland, here's this olive-colored form. Same thing. Down in Colula, here's a gray-brown form. In the Paluma Range, just north of Townsville, there's a white one. And the thing that you got to keep, I want you to focus on here, is that the tube colors are pretty much the same. They all bruise. The mushrooms themselves bruise brown. They all have very similar macroscopic features. Down around the border ranges near Springbrook with New South Wales and Queensland, here's an orangey yellow one. Again, the same group. Now, having the opportunity with Neil Bowser and Richie Robinson to explore some of the Jarrah bushland in Western Australia, here was this yellow viscid thing. It loses some of its color with age. As it turns out, it's the only one of this group that had been described as a species and it was described by Ra Watling. And he put it in the genus Rubinoboletes. Again, it's not that genus at all. And it got its species name because it has bean-shaped spores. And in the lower left, the other thing that's characteristic under the microscope are these sterile cells called cystidia that have these oily globule-like structures on the inside. And if you look next to this one cystidium, here's a long bean, but it's attached to the sporogynous cell called a basidium right there. And the DNA tells us it's not a Rubinoboletus. Rubinoboletus in this phylogenetic tree is way off the map. It's not even here. But here's a beluioid thing from Fraser Island, one of the orange jobs, and it's related genetically to the genus Tylopolis, and Phelius is the type species of the genus. That's the species upon which the genus Tylopolis has been described. So what I've called here Balui is not really Balui but in the broad sense, okay? Another genus that's common in Australia is Austroboletus. And up to this point, I can estimate there are probably 17 species, 5 of which are still undescribed. One of those, which we just described, Asper, here in the picture, with the help of Katie Syme in WA, we were able to describe just this past December. So I'll just show you a few pics of some of these iconic Austroboletus species that we know about in Australia. The first one to have been described was Latumnosus, described from the vicinity of Brisbane, sent by Bailey, I think it was, to MC Cook in Britain. And it has very distinctive macromorphology. It starts out wet and sticky, dries out, but it has spores that are sort of almond-shaped, and they're very heavily ornamented with a scanning electron microscope. You can see that in the lower right. One which we found in 1992 on that trip with Neil and Roy Young et al. Up in northern Queensland, we found this Austroboletus and had no idea what it was. And actually I thought I had 2 or 3 different ones because it changes over time. When it's young and fresh, it starts out red. As it ages, it gets orange. And if it sits around for a while, it's completely yellow. So that's why we called it mutabilis, which means changing. So we called it Austroboletus mutabilis. And the interesting thing about the spores is completely unlike other Austroboletus spores that we know about, because it has this naked area on the spore called a plage, where there's no ornamentation, okay? Another Austroboletus, which was first described from New Zealand by Greta Stevenson, is Austroboletus niveus. Later, Roy Watling described Austroboletus eburneus from Queensland, and it turns out they're the same thing. They're white and dry, but they have a little bit of stickiness to them. And I tell you, don't eat it. Don't touch your eyes after you touch it because it'll burn like crazy. The thing about the spores of niveus is that it looks like fine sandpaper when you use a scanning electron microscope. Under a light microscope, that very fine ornamentation is extremely difficult to see. And Roy Watling described Austroboletus occidentalis from Western Australia. And so far as we know, it's the only place where it grows. One bit lacking from the original description was the spore morphology, or the spore ornamentation. He neglected to mention that the plage of the spores in occidentalis are naked. There's no ornamentation there. And I thank Katie Syme for sending me the material and the photograph for which I could make the SEM and document this species. And having the opportunity to work with other folks in New South Wales, Ian Dodd Teresa and John Vanderhoel, and Pam O'Sullivan, we discovered this other delicate, fluffy, sticky Austroboletes. And it's not very big, it's very delicate. And it's got this white fluff that breaks up to form these, to show these pink pigments in the cap. And the spores are extremely ornamented, as you can see there on the right. And we only described this one a few years ago. All right, let's move to another genus that's iconic in Australia, and that's the genus Boletes. The first species of the genus described from Australia was the one called Ananiceps. That's the top of the list. So trying to figure out what Ananiceps was all about is what I call forensics, mycoforensics. If you go back to the original description by Berkeley in 1873, nine or 9.5 words in Latin are the description for this thing called Ananiceps. What the heck does this mean? Well, if you translate the Latin, it comes out pileus convex, da-da-da-da-da. And the locality, Wangaratta in New South Wales, and the spores were huge. 80 years later, Rolf Singer looked at that specimen and he revised the spore size, and he described the spores as having wings, but the wings of the spores weren't striate, meaning at what I know now, meaning they don't have cross bands across those wings. And based on Singer's classification at the time, this belonged in the genus Boletes. So, walking around on Fraser Island for 3 years with Nigel Fechner, we came across a bolete there on the lower right that had these thick, large flocosquamous warts, just described by Berkeley. And the spores from that thing had wings that were not striate. Further collecting showed us that younger material of this Boletes ananiceps, there was pigment there, pink pigment, but it was underneath this ochre-colored layer of mycelium, felty layer on the surface of the cap. And as it aged, that ochre-colored layer expanded a little bit, but the pink color stayed underneath. And eventually that expanded into the flocosquamous warts. But that's all we knew about what this Boletes was until Nigel and I were able to investigate it further. And then we found, it has a veil, and the tubes and pores stay blue-green, and the stipe is white, and the stipe stains brown when you touch it. When you cut the bolete longitudinally, it stains blue, and it stains blue really fast. But if you're careful and you do what I call a quick cut and look, you can see that the actual color of the flesh is yellow. And that's where the blue staining is happening. Further down on the stipe, there's no blue, it's white. And down here, it stains brown. So this is another characteristic for this species that we call Ananiceps. And it's completely unlike the New World bolete called Boletes ananas, which is the type species of the genus. Here you can see the spores have the cross striae on the wings. Well, poking around on Fraser, Nigel and I, the first time we found this other bolete, and we had no idea what we had. We looked it up in Corner's book on boletes in Malaysia, and it turns out that he had collected this thing in 1931 in Singapore, but hadn't described it until 1974. And it turns out to be another Boletes with winged spores with cross bands, but there's no pink in it. The stipe stains brown, but it gets it can get these flocose, thick squamose warts, but it doesn't have any red or pink in it. And again, when you cut it open, you can see the bluing up here. And if you do a quick, what I call the quick cut and look, you'll see that it's yellow before it stains blue. By the time you cut this and lay it up for a photograph, you can't see that the flesh was yellow. But again, further down, the context is white and stains brownish. Another iconic one in Australia is what's called Emodensis. And this one, when it's fresh out of the oven, it's red, red, and red, except for the tubes. The stipe's red, the cap's red, but it has really fine scales on the cap. And the spores have wings that have cross bands. The thing about Emodensis, besides that, is that it stains blue all the way down the stipe. And as I said, the quick cut and look will show you that the stipe is also yellow before it stains red. That's another key characteristic for Emodensis. Now, on a trip to Queen Mary Falls in southern Queensland with Nigel, we came across this bolete, a very deceptive one, and that's how we gave it its species name, because it had very fine scales that had some red, but they were also white. There was some red in the stipe, and they looked like that one in the lower center was developing some squamose scales. So what the heck do we have here? And it turns out, based on the DNA analysis that we did, it was a completely different species. And that's why we called it the Deceptivus. And when we did the scanning microscopy of the spores, we found these holes that transversed those wings inside the wings. And doing my forensic literature search, I found that Jacqueline Perrault in Paris had done some transmission electron microscopy, and she showed that spores of another species, which has these striated wings, had these lacunae inside the wings. So what we have, when you look at the spores with a light microscope, is that you have the light refraction based on the solid part of the ribs plus the lacunae, causing this light refraction to give you the idea that there are cross bands on the spores, but they're actually inside the ribs of the spores or wings of the spores. And one of the target species of fungi map is Boletes obscuricoccineus. And as you can see in this photo, it assumes all kinds of different macro morphology in terms of colors, stipe, ornamentation, etc. And the spores clearly show you that it's a Boletes. Now I remember last month Tom May was talking about populations and how things might vary across different geographical ranges in Australia. Well, just based on what we have seen for this target species in Queensland, the ones up north in Paluma, Davies Creek, will have a different genetic structure than the ones we found in southern Queensland on Fraser Island. When I was working here, working here in the northeastern United States, one of the iconic boletes was called Boletes chromapes when it was described in 1874. Turns out it belongs to this genus called Harria. But there's nothing else like it in the United States, in North America. Pink cap, pink dots on the stipe, chrome yellow base to the stipe, but sometimes that stipe can also have a reticulum. While collecting in Queensland and New South Wales, I found the same thing, reticulated stipe. I say, so do we have an Australian Harria growing in Australia. And work that Neil Bowser had done with another North American mycologist, they described specimens from northern Queensland in the genus where this thing belonged at the time. And they called the one in northern Queensland, Queenslandianus. And you can see that it's got the little pink dots, bright yellow stipe, pink cap. And the ones that had a reticulum were called Palomanus, based on specimens collected from Paluma. It turns out that this group of stuff, these chrome-footed boletes, belong in another genus, which we described as Australopilus, completely different in the upper part of this gene tree, that Palomanus and Queenslandianus were all the same thing, and that true Harria from the US and apparently from China is completely separate. But embedded in the middle, here is this thing called Royungia, this truffle. And it looks like this. It turns out that the chemistry of Royungia matches the yellow pigments in the foot of the stipes of the other ones. So, newbies, let me get those quickly out of the way. There's a lilac thing that occurs, and you can see if there's some kind of obstruction across the top, you won't see the pigment. The other thing about this bolete is you can't see the reticulum on the stipe because the reticulum is white. If you bruise it, immediately it turns blue-green. Later, that blue-green turns to brown. Same on the stipe. Now I go diagonally to this thing called the true porcini with Tylopolis spores. This is a thing we found in 1992, and I said, wait a minute, what the heck's going on here? They said, and you see that blue stain on the stipe. In northeastern North America, there was a species described by Charles Peck, same one who described Bolete, and he called it Boletes separans. It turns out separans is a true bolete related to porcinis, and it behaves exactly the same. In Eastern Australia, known from collections up around Davies Creek in the Atherton tableland, there is this bolete, and it also occurs around Newcastle in New South Wales, which we only discovered back in 2017. Nigel found it, as a matter of fact. And when you put ammonia on the stipe, you get this turquoise green reaction. However, the spore morphology and the tube morphology is exactly like a true Tylopolis. So we have this other thing that needs to be worked out. Now let me move on to the upper right where it says Chalciporus ? Had the opportunity to work with Heidi Pritchard when she was doing an honors study in University of Newcastle, and we thought we had a report for a native species of Chalciporus in Australia, not the imported one that came with Amanita muscaria. So we're still investigating this to see if indeed this is a true Chalciporus or not. And that's still a work in progress. Down in the lower left, there's this thing that I call Porphyrazi in quotation marks. Well, it's a genus that we don't know yet. I just made one up. But the species is known. The species is called cyanogranulifer. And I'll show you why in a second. My first inclination after collecting this about 45 times was that it seemed to match a species described from New Zealand called Porphyrellus bruneus. Well, it's not that species. It's not that genus either. It turns out that Roy Watling, based on the generic concepts he was using at the time, he called it a Tylopolis. And he called it Tylopolis cyanogranulifer. And the reason he did so was because if you mounted a portion of the pileus surface in ammonia or KOH, you found that the hyphae of the pileus surface had these cyanogranules. And this is very easy to see, even before you put it under the microscope. When you take the tissue and you mount it in a hydroxide, KOH or ammonia, you can see the blue-green reaction happening just in the pool of the mounting medium before you look at it under the microscope. But it's not entirely clear because it could, that is, there appears to be another group of at least one or two species that have the similar macromorphology of cyanogranulifer that don't have those blue-green granules in the pileus. So we're still trying to sort this out. And Naveed Davudian, who's now at Melbourne, is going to help me sort this out. So, take a deep breath. Where are we? Clearly, the ecological lifestyle of boletes is paramount for bushland survival. They bring water and nutrients to the tree from poor soils. They explore more soil volume to get that stuff to the trees. And the more that you explore, the more you're going to find. And it's not just, having more eyes looking around in the bush. It's going to bring out new discoveries. And those new discoveries are going to expand and refine the census of what we know about that occurs on this planet, the diversity on this planet. And because up to this point, our bias about what boletes are was based on a North Temperate view, because that's where things started out in Europe in mycological taxonomy. And so I want to say that more exploration, we're going to find heaps of examples in Australia that's going to build on what we know in terms of mycological diversity. And finally, I have to say, I have to thank Teresa LaBelle and Nigel Fechner, two mentors who helped me tremendously get my feet on the ground and try to understand how to do work in Australia, how to truffle for things in Australian sand. Nigel for keeping snakes away from me, but more than that, showing me where to go, how to find new habitats and new interesting places. And truly mentors for teaching me and helping me get along in Australia. And lastly, I can't show pictures of everybody, but these are all of the other people in Australia who have helped me get through and understand what was going on in that part of the world. Thanks, Susie.
Thank you, Roy.
Pleasure.
Taking me right back to my bolete workshop, and I'm remembering all these things now that I'd forgotten before. Very exciting. We need you to do it again.
Well, one day I hope so.
Yeah, well, yes, post COVID and post the rest of your shop.
Right.
Few more questions. Vanessa has asked, how many species of boletes in Australia are also found overseas in other countries?
Well, there are some in New Zealand that are also in Australia. I can't comment specifically at the moment, but I'm reviewing a paper for something that might also occur in Southeast Asia. Oh, I do know that some of the things I've seen in New Guinea also occur in northern Queensland. Emodensis was described from Sikkim in the Himalaya. What did I say? Decilians was described from Singapore. So there are some things that seem to have crossed Wallace's line from Southeast Asia into Australia, and that must have to do with the movement of plate tectonic, continents moving and slamming into each other, and then things migrating back and forth. And as I said, boletes are mycorrhizal, but they don't have to be very picky about which tree or species or genus they might associate with. It could be oaks or lithocarpus in New Guinea or dipterocarps in Southeast Asia, but they've crossed over and now are with gums and alocasarina in Australia. Another example of that is that Austroboletus niveus in New Zealand. It occurs with Nothofagus, the beech, but in Australia, no, it's with Myrtaceae and alocasarina. So they can swap. Yeah. As long as something's appropriate, I suppose it doesn't matter.
Sounds like there's something connected slightly between them.
Yeah, it could be.
Katrina has asked, are you coming back to Australia? Well, I think we've established that you want to come back. It's COVID and travel permitting.
Right. As soon as, number 2 vaccine in a month, or less than a month, 3 weeks away, and travel restrictions are lifted, I'll come back. And I'm retired, so I don't have to punch the clock anymore. So.
Excellent.
Yeah.
Peter's asked, Amanita muscaria is relatively new to WA, to Western Australia.
Oh, okay.
And says, I have not seen a Calceoporus paratus here. Have you seen it here?
In
you didn't
WA?
cover the paratus, did you? In WA?
I haven't seen, no, I was only in WA one time. Well, no, twice. The first time in 2006 with Neil, I didn't see it then. And then the last time was 2011. I didn't see it then either. But the first time when I saw it was in Tasmania at a FungiMap conference in 2005. And we had driven we're out in the Tarkine, and there wasn't a pine anywhere. And here's Amanita muscaria. Everybody's getting out to take pictures. And so they took pictures of the Amanita muscaria, but they neglected to see right next to it was this little tiny Chalciporus piperatus. In New Zealand, I've seen it all over the place. I haven't seen the Amanita. Well, when I've seen Chalciporus, I didn't see the Amanita, but I've seen both. So it's there. They may not be fruiting at the same time.
Okay. Sonia's asked, have you had a fungi named after you yet?
Yeah, finally. No, I shouldn't say that. Not finally. Yeah, Mike Castellano, the fellow that I showed on
the left of the picture?
Yeah, that's got cassia named after him. He works on this group of truffle-like fungi, which are, they're truffle-like, but they seem to be related to stinkhorns. That's Mike's specialty. And in Chile, Argentina, and the Antarctic beech forest, there's one there, and one of these things. And so he called it Halingia. So I have a stinkhorn truffle with my name as the genus.
Infamous name.
Yeah, I guess. Anyway, but
very good though.
Yeah, I've never seen the fungus, so I've just seen pictures of it.
Okay, this is a bit of a wider question. Shane's asked, how can citizen scientists help mycologists describe or find new bolete, and can you describe how an herbarium collection is made? Yeah,
in that link that I sent to you and Sophie, there's some PDFs on my website that describe how to properly make specimens for scientific study. And there's some, what do I call them, cheat sheets? I'm not sure that's right. There's some illustrated sheets one for guild mushrooms, another one for boletes. I didn't write them, but they give you an idea of what to look for when you're describing or writing notes about what this fungus looks like. But the key is that I remember one of my mentors way back when I was a graduate student, and his name was Alexander Smith. And he said, I didn't come here to photograph, I came here to collect. And so he would fill his basket, then he would spend the rest of his daylight hours, 4 p.m. on, working on the collections. And that's often why you'll see some of my pictures look like portraits rather than pictures out in the bush. Because he went to collect everything he could find during the daytime, and at nighttime he would take the pictures and write the notes. So it's a matter of, you can spend all your time taking pictures in the bush, or you can collect like crazy and come back and spend all your waking hours till 2 a.m. working on writing them up and describing them. But the key is, if you're going to collect something to make a specimen for science, to deposit on herbarium, is to work on it as soon as you can, because over time, the thing will start to collapse, start to decompose, start to fall apart. So what you saw in the bush isn't what you got when you came home and sat down at the table. And so the key is to work on it as fast as you can, or as soon as you can get it. So, but anyway, look at those links that I sent you, and if people have questions, they can always send me an email.
Yeah, we'll actually send out that link when we send out a copy of the talk for everyone.
Okay.
Just thoroughly enjoyed this. Thank you very much for all involved, from an anonymous attendee. But one more question: are boletes common in arid environments? And Malcolm's asking this.
Well, I guess it depends on what your concept is of an arid environment. When I first went to Queensland, and in '92 with those folks, and we're driving around, and I say, why are we stopping here? There's no mushrooms that grow here. But indeed there were, and you just had to shift your concept of what's a habitat that's appropriate. For boletes, you have to have trees around. And so, I can't imagine, walking around in the outback outside of Alice Springs without if there are no trees around, you're not going to find boletes. There could be truffle-like boletes there, but I'd never explored it. So, yeah.
Next time you come, we might have to go out and have a look.
Okay, that sounds good.
A little note to you from Pam O'Sullivan. Thank you so much, Roy. Wonderful talk. Great to see you.
Thanks, Pam. Thanks so much you did for me. I appreciate it.
All right. Thanks, Roy.
And send emails if you have them and have any questions.
Yep, your time's valuable, and sadly, time has definitely run out for us.
Okay.
It's been a great talk. Loved it. Reminded me of so much. For those of you who still have questions, we will send out the link to Roy and to the video, the recorded session today. If you have any answer, that'll get you the answers to any extra questions you have. If you'd like to join FungiMap, purchase books, or just have a look, please go to our website, fungimap.org.au. Also, if you've enjoyed our webinar with Dr. Roy Halling, you may also like to book next month's Funginar with Dr. Jordan Bailey, the Curator of Plant Pathology and Mycology Herbarium in Orange, New South Wales. Information on that will be available at the end of the week, and Sophie will send that out. She will be speaking about the importance of collections and support FungiMap's work and research data. Anybody who had those questions relating to Roy and Bolete might want to follow that one as well. So thank you, everyone. Good day from FungiMap, and see you at the next Funginar.
And just before you go, I just wanted to also thank you, Susie, for all the work that you've been doing, putting into this webinar series, organising all the speakers and making it all happen. So yeah, thank you. And yeah, huge thanks to you, Roy, for so generously giving us your time today.
No, thank you both. I appreciate it.
Thanks,
And
Sophie.
everybody else.
Yeah, thanks, Roy.
Okay.
Always good to see you.
Likewise.
All right. Thanks, everyone.
Cheers.
Bye.
Bye.
Bye-bye.